Start video. Yeah. So welcome everybody. So I'm Professor Fiona Russell. I'm the Director of the Child and Adolescent Health PhD program at the University of Melbourne, and also I lead the Asia Pacific group at MCRI. And so it gives me great pleasure to co-chair this session with Professor Steve Graham. This is our first and inaugural, in fact, Asia Pacific vaccine research seminar series that we've started, and just a little bit of background. I won't take up too much time, but just to let people be aware that so we've got a DFAT Australia Awards Fellowship that's come from our partners, sort of request really from our CRE forum that we had last year. And from this fellowship program that we've got, it's really to help us establish an Asia Pacific vaccine research network and to increase vaccine research capability for policy decision making in the region, and also an opportunity for partners in the region to share their learnings and to sort of teach each other as well. So we're doing that in partnership with Professor Itop from the Universitas of Gadjah in Indonesia, and our fellows are from all over the region, as sort of listed there. And so part of that training is some coursework here at the University of Melbourne for the fellows to come to Melbourne, and we'll have some workshops and masterclasses using immunisation case studies, as well as going to Indonesia as well and undertaking some workshops there. And we'll be having sessions on, research and how to do the research, particularly with regard to research that's needed to inform policy, and also using that research to be able to then make recommendations for immunisation policy. And so this webinar really is part of an ongoing professional development for vaccinologists, vaccine researchers, NITAG members, or Ministry of Health and Immunisation staff. So I'll now hand over to Steve, who's going to lead our first webinar seminar series, which is on TB vaccine. And just before we get going, I think Nigel wanted everyone to put on their video, if you can. And we'd like to take a photo. I think Nigel wants to see your eyes and make sure that you exist. Yes. Okay. So I might have to do two photos, actually. I'll do... Okay, that's the first one. Oh, there's 3 pages on my screen. Hang on. Oh, not everyone's got their camera on, though. Maybe you can't. Anyone else can put their camera on, please do. Otherwise, you'll miss out on the inaugural event. Okay, and one more. Okay. Oh, this is a blank screen. All black here. All right. Okay. All right, no worries. Over to you. Thank you very much. Over to you. Thank you, Steve.
Thanks, Fiona. Thanks. I think this is a great initiative. In our region, I think tuberculosis is a major ongoing challenge for infectious disease, causing morbidity and mortality in adults and children. And a vaccine has been, a very effective vaccine, I should say, has been elusive. So look, the first speaker, it gives me great pleasure to introduce, is Dr. Trisasi Lestari, known as Sasi, who's going to give us an update on the burden of TB and multi-drug resistant TB in the region. And Sasi has recently completed her PhD studies on improving TB case detection and prevention in Papua provinces of Indonesia. She's been involved in TB work for a very long time, is a public health specialist, and is with the Centre for Tropical Medicine in the University of Gadjah Mada in Indonesia, and also with Charles Darwin University in Darwin. So over to you, Sasi, to give us an update on the burden. Thank you. You can see the slides. Can't hear you, Sasi. Are you muted or? You're still muted.
Yep, sorry, I'm still muted. Okay. Can you hear me now?
Yeah, good. Thank you.
Sorry. Yeah. Thank you, Steve and Fiona, for the opportunity to present in this important forum. Actually, I'm certain that the audience here are well aware of the significant burden of TB globally, as well as in Asia Pacific. Found it challenging actually to present a topic that's familiar to all of you, and probably many experts in the audience have better knowledge than I am. But anyway, I will try, and I hope you gain something new from this presentation. So, vaccination, when used together with other preventive measures, has the potential to reduce the incidence of communicable disease. And we all know that BCG has been used for more than 100 years, and it is the most commonly used vaccine worldwide. BCG immunization coverage in all WHO regions are actually relatively high. In Southeast Asia and Western Pacific region, the coverage are more than 90%. It has proven its efficacy in children. So in a recent meta-analysis by Martinez et al., they showed that the overall effectiveness of infant BCG vaccination against all type of TB was only 18%. But when stratified by age, BCG vaccination only significantly protected against all tuberculosis in children younger than 5 years old. It is effective to prevent primary TB disease or disseminated TB, such as tuberculosis, meningitis, and miliary disease. The efficacy of BCG is waning after a few years, leaving adults susceptible to TB infection. So this graph shows that TB incidence increase in adolescents and adults, and that the incidence is higher among male groups. So the study by Martinez et al. Also showed that the BCG vaccine was significantly protective against tuberculosis among female participants, but not among male participants. So this could be partially explain the situation among other contributing factors. But I think we all agree that we need new TB vaccines that can provide better protection for all age groups, including vaccine that can prevent TB in adolescent and young adults who are responsible for TB transmission in the community, as well as a vaccine that can provide equal protection for male and female population. So I recently found this article, and for me, it's quite interesting personal view article written by David Doidi and Marcel Berg that probably some of you also already read this article. They argue that the contemporary annual risk of infection might underrepresent the true annual risk of infection. So ARI estimate primarily derived from cross-sectional TST or IGRA surveys. So for example, this figure illustrates the population structures and projected tuberculosis status by age in India in 2016, assuming an annual risk of infection of 1.5%. In this scenario, 75% of incident tuberculosis reflect late progression. So Doudi and Berg, they propose a higher annual risk of infection of 2%, increasing to 5% in older age group. And recent infection will account for more than 73% of all TB incidents. Studies of household TB context suggest a higher annual risk of infection immunity than in the home. Latent TB infection in the household is estimated higher than the latent TB infection.
Oh dear. Sassy's frozen. I'm not sure whether she can hear me. Should we just wait a little bit? What do you think, co-chair?
Yes, I wonder, I don't think we've got her slides either. I don't have them, but yeah, maybe wait to see if she can come back on. Was her video on? Maybe oh, here she is. She's back.
Yeah,
She's back,
sorry.
okay.
Maybe my internet is dropped. Where are we previously?
That's okay, Sassy. You just covered the
Continue.
slide, I think. Yeah.
And maybe just turn your video off just in case. So
Yes.
for now, yeah.
Yeah. Sorry. So this finding, the study from Dodi and Bear study is actually similar with the finding from the Penabulu STPI consortium that organized large scale of TB contact investigation project in Indonesia. So this is an unpublished data. In 2022, more than 4 million contacts were investigated, and TB screening was conducted based on TB symptoms, and they are performed by community health cadres. So individuals who reported TB symptoms or healthy children under 5 will be referred to a nearby health facilities for further TB examination. And among household contacts who have sputum tests, 3.9% were bacteriology confirmed TB, compared to 9.8% among community contacts. The proportion is higher if we included all contacts who visited the health facility for TB examination and include contacts with, and if we include contacts with chest X-ray positive or clinically diagnosed as child TB. So the proportion will be 4.4% among household contacts and 10.8% among community contacts. So this result from the Penabulu STPI consortium support the argument that annual risk of infection in the community is probably higher, and therefore will also have implication for how we should conduct TB contact investigation in the community and TB screening or other public health efforts to eliminate TB. The obstacles and in the introduction and use of new vaccines are often more pronounced in the countries where the disease burden is greatest, and the factors that contribute are not only limited to economic power of the countries. So from an article by Levin et al., they propose a policy framework for considering the issues and challenges to accelerating new vaccine adoption. So they propose 3 phases of new vaccine introduction. First is to establish and organize evidence. Second is to establish supportive global policies. And the third phase is to translate policies into local action. And for this session now, I'm going to highlight the burden of TB in Southeast Asia and in Western Pacific region, which most of you probably already familiar. The WHO estimates that about 10 million people develop TB every year, and 85 to 90% of these people live in one of 30 high burden countries, and 12 countries are located in Asia Pacific region. 5 out of 7 countries that accounted for 2/3 of global MDR-TB cases in 2021 are also located in Asia Pacific. We also have learned that disrupted access to healthcare services and active case finding activities during the COVID-19 pandemic have immediate impact to TB program, causing setbacks in countries that were making progress prior to the pandemic. And the top 10 countries that accounted for more than 90% of the global reduction in case notification of people newly diagnosed with TB in 2020 and 2021 are also mostly located in the Asia Pacific region. So overall increase in TB incidence between 2020 to 2021 has also led to a rise in the estimated number of incident cases of DR-TB in 2021. The proportion of DR-TB cases is significantly lower among new TB cases and almost 5 times higher among previously treated TB cases. So this fact actually raises a question: is there a potential for therapeutic vaccine to reduce the incidence of DR-TB among previously treated TB cases? Reduction in the TB case finding due to disruption of TB services, increased transmission of TB infection in the community, delay of diagnosis, or failure to access appropriate treatment may cause a fatal outcome. And the WHO estimated that there is an increase in number of TB deaths. In Southeast Asia region, it is estimated that there are about 800,000 deaths among individuals with TB and about 125,000 deaths in the Western Pacific region, which contribute to about 58.4% global TB mortality. So time is of the essence for this effort. Every day that is lost in the introduction of new technology, including vaccine, means that many people with literacy suffer. And in the context of TB, assuming that approximately 10 million people develop TB every year, then every day of delay in full access of TB vaccine means that minimum of 25,000 individuals are exposed to higher risk of contracting TB. So I'll end my presentation here. Thank you all for your kind attention. I'd like to acknowledge the Mensi School of Health Research, Center for Tropical Medicine at Universitas Gadjah Mada, and the Foundation for Papuan Health and Community Development in Papua, and also thanks to the Murdoch Children Research Institute for the opportunity, and the Penabulu STPI Consortium for sharing the TB contact investigation data. Thank you very much. Back to you, Steve.
Thank you so much, Sasi. Thanks for a very succinct presentation. And I think you've introduced nicely the burden, but also some of the potential for vaccines. If it's okay, because of the Zoom, I think it may be easier, not sure what you think, Fiona, to do the question and answers discussion all to the end.
That sounds great, Steve.
Yeah, so, and by all means, people put your questions in the chat. Welcome to all of those who are still joining. I see folk from Fiji, from Cairns, currently doing PhD in development of new vaccines. So this is fantastic to have already an audience of up towards 3 figures. So that's great. So now we have an excellent speaker, Professor Nigel Curtis, and Nigel's a colleague of ours here at the Melbourne Children's and Global Health Group, and he's a professor of infectious diseases, a pediatric infectious disease specialist, clinically active at the Royal Children's Hospital. He leads a lot of research in infectious diseases, including over many years, research into BCG vaccine, its limitations, its immune response, its protectiveness against a range of diseases. So he's an excellent person to talk to us about some of the limitations of BCG and the challenges of developing a new vaccine. Over to you, Nigel.
Good afternoon, everyone, and thank you, Steve and Fiona, for the opportunity to speak to you today. Now, you might ask yourselves, how did they persuade me to talk to you today? And the answer is that Fiona promised that I would get at least 3-figure additional followers on Twitter. Of course, it's no longer Twitter because the bird has been killed. So this is bang up to date. It's now X, or however we pronounce that. So please make sure you join up before we finish this talk. So what are the challenges of developing a new TB vaccine? Well, number one, I would contend, is that we already have a very good TB vaccine that's very hard to beat, which is, of course, BCG. In fact, it's the only vaccine we have that protects against TB. And this map of the world, which is distorted by the number of births in each country per year, you can see that of most of those births, 120 million of the 130 million each year, occur in areas of the world where there is still a high prevalence of tuberculosis, and infants in those areas receive BCG shortly after birth. And as you've heard, this is the most widely used vaccine worldwide. Now, why am I obsessed with BCG? But the reason is, if you look at Wikipedia, where this talk comes from, you'll see that the vaccine was invented by Professor Nigel Curtis when he was a medical student. Now, many of you will know that I've used this slide so often that I've actually started to believe this. And in fact, if you look on tuberculosis BCG vaccine on your Instagram account, you'll see it says, "So pleased that Professor Nigel Curtis invented BCG, #vaccine, #genius." And it's not just me who now believes this. You can see this recent tweet from Elon Musk. I'm incredible that Professor Nigel Curtis invented #BCG vaccine. Even I have not created a vaccine. So this really has grown legs. But of course, this is fake news because the vaccine was created by Albert Calmette and Camille Guerin more than 100 years ago. And what they did is they took a Mycobacterium bovis and they passaged it 231 times over a 13-year period until this bacteria became avirulent. And when they were fairly certain that it was avirulent, they used it in animal experiments to show that it could protect against tuberculosis, initially in cattle. And I think you can see here, these are from the days when you didn't need statisticians. You can see which is the control very easily. Now, after this has been shown to protect against TB in cattle and other animals, it was then trialed in humans. And you can see here these children lining up for their BCG. And I've always thought this chap here looks very worried. And the reason he looks worried is that he actually saw some of the early studies of some of the BCG strains before they'd been passaged a sufficient number of times. So there were very large studies in the 50s. You can see this study had over 50,000 participants. Just think about that with paper records, which showed that this vaccine was highly effective in protecting against tuberculosis. And this nice graphic, which shows the number of deaths that have been prevented by vaccines, you can see that BCG has saved as many lives as any other vaccine over over at least the last 25 years. And you just saw this paper already, but this nice meta-analysis by Jason Andrews and colleagues shows that in children, this vaccine is highly effective at preventing deaths. So you can see there the adjusted odds ratio, 85% protection against death in all ages, but an 80% protection in those under 5. This is a very effective vaccine preventing the severe forms of tuberculosis that affect infants and young children. Now, after they created this vaccine, Kalmet and Guerin distributed this vaccine all over the world, where it continued to be subcultured. And as a result, during culture on different media, it continued to mutate and various genetic changes occurred in the strains. And so, in fact, there isn't just one BCG, there are multiple different BCGs. And this continued until the early 50s when seed lots were created by lyophilization. So we now have different BCGs which are named after the place that they were originally sent. And in this study, which looked at the global use of different strains, you can see those regions in red, which are where there are very high prevalence of tuberculosis, use these 3 strains, which are the most commonly used worldwide. So that's BCG, Denmark, Japan, and Russia. And it was in 1999, with the advent of DNA technology and microarrays, that it was shown that these different strains of BCG do have major differences in their genome. They all miss RD1, which is the region of difference 1, which is what makes them avirulent, but they also have many other mutations. And so it's been suggested that we should actually call these different vaccines rather than different strains. And when Nicole Ritz was here, many of you will know, during her PhD, she randomized infants in Melbourne to one of those 3 most commonly used strains. Now, what she did in this review was showed that those different strains vary both genotypically, phenotypically, and even in the number of CFU per dose. And certainly there's many studies showing a difference in protective efficacy in animals. But the big question is what happens in human. So in randomizing 2-3 different strains in infants in Melbourne, and it's a bit sad to kind of summarize someone's PhD in kind of 3 years of work in one slide, but essentially what Nicole Ritz showed was that there is a difference in the immune response that is induced by these 3 different strains. But the big question is, does this translate to clinical differences in clinical protection against tuberculosis? And unfortunately, there has not been any randomized controlled trials using those strains. But there is some clues from observational studies that it does matter. And this is my favorite one from Kazakhstan, which by looking at different periods when different strains were used, they were able to show that BCG Japan was far superior in protecting both clinical and culture-proven TB than both BCG Serbia, which is really BCG Russia, one strain of BCG Russia, and BCG Russia itself. And all 3 were more effective, of course, than no BCG at all. So we have this indirect evidence that strain really does matter. Now, when we talk about protection, we're normally talking about protection against tuberculosis, but BCG has other properties. It also protects against other mycobacteria. It has cross-protective effects. In this systematic review by Petra Zimmerman, she showed that there's very good data showing that BCG protects against leprosy, lymphadenitis caused by Mycobacterium avium intracellulare, and also Buruli ulcer caused by Mycobacteria ulcerans. But over and above mycobacterial protection, BCG has non-specific or off-target effects, protecting against bacteria and viruses other than TB and related pathogens. So in this seminal study from West Africa, it was shown that survival was greater in those babies that had BCG at birth than those who didn't. And that difference in survival is greater than can be explained by protection against tuberculosis. And in fact, this is protection against the kind of things that kill infants in high mortality countries, such as pneumonia and sepsis. And there are other studies, for example, more recently, this study from Uganda, again, showing a reduction in blue in the incidence of doctor-diagnosed infections in the first 6 weeks of life. Now, we now have some understanding about how BCG does this. And one hypothesis is that this is due to trained immunity, which is the recently understood property of cells of the innate immune system to have memory. And in fact, it's thought that perhaps it's trained immunity that actually contributes to the BCG-induced protection against tuberculosis. Now, when we looked at this forest plot of protection by BCG from this systematic review, what I didn't show you was protection in the over 15s, where there's very little protection in this meta-analysis against death from BCG. And when we look at just tuberculosis disease itself, BCG is not so effective. So slightly tongue-in-cheek to say that we have a good TB vaccine that's hard to beat. We certainly need a vaccine that is better than BCG to protect against TB, and particularly in older age groups. And there are other problems with BCG that we don't understand. And one of them is why there is such varying protection in different parts of the world. And in this, again, very famous study by Paul Fine, published in 1995, showed that protection was greater the further away you are from the equator. So there is this correlation with latitude, and least effective in areas around the equator where there is a lot of TB. And it's really still not understood what explains that phenomenon. So what is the challenge of developing a new vaccine? Well, one of the challenges is that both humans and TB have co-evolved. And so tuberculosis has been found in mummies that are 2,600 years old. But in fact, TB has been thought to have been around for at least 9,000 years. And it's been shown that both MTB has evolved over that time together with the human immune system to coexist. And so you're really fighting against 9,000 years of evolution when you're looking to prevent tuberculosis. And the problem is that we don't understand what it is you need to protect against TB. We don't have any correlates of protection. It was always thought that it was T cell immunity that was most important, and particularly Th1 cells that are producing multiple cytokines. So polyfunctional cytokines producing gamma interferon, TNF alpha, and IL-2. And certainly in animals, that is the case. Now, in this, one of my favorite studies of all time from Willem Hannekom in Cape Town in South Africa, reported in 2010, nearly 6,000 infants were vaccinated with BCG Denmark at birth. And after 10 weeks, they had blood taken, and a very large array of biomarkers were tested and banked away. And those infants were followed in a very high TB prevalence area for 2 years to see who developed tuberculosis. And there were 29 infants who developed TB who were therefore deemed to have not been protected by their BCG at birth. Now, there were 55 babies who were household contacts of adults with smear-positive TB who did not develop TB and therefore were likely protected from their BCG at birth. And what they then did was a case control study to look what was the difference in the way those infants responded to their BCG when measured at 10 weeks, to try and understand what were the biomarkers that are indicative of protection. And unfortunately, what they showed was that there was actually no difference in those polyfunctional CD4 cells that were thought to be so important, and in fact are important in animals. And in fact, no biomarker was found to be different in those that were protected and those that were not protected. And that's consistent with this big trial by Helen McShane and colleagues of the MVA85A vaccine amongst nearly 3,000 infants. And what they found in that trial was that BCG did, in fact, increase the number of these polyfunctional T cells. So it certainly had the immune response that was hoped. But in the clinical trial, in the primary outcome, there was no difference in protection against tuberculosis. So we almost know more about what is not indicative of protection than what is. And there is still nothing that we can measure in someone to tell you whether they are whether they have any protection against tuberculosis. So what other parts of the immune system might be involved? Well, we've done some work here showing that perhaps non-conventional T cells and NK cells may be important, and similarly, the humoral immune system may also play a role. So lastly, the other challenge of developing a new vaccine is, of course, that TB is a disease of poverty, and so it may not be as attractive as other vaccines to the pharmaceutical industry. And this figure illustrates here the top 10 most populous countries. And the red bar shows the TB prevalence and shows that it is inversely proportional to the GDP, shown in the blue circles. So I'd like to end here by having shown you those challenges, but just with a plea to everyone listening who's working on developing new TB vaccines. And that's that we have to be mindful that any new TB vaccine, as well as protecting against TB, still has to have off-target effects, because if we remove BCG and replace it with something better at protecting against TB, but that does not protect against other infections, there may be a paradoxical increase in all-cause mortality. And this really this understanding that BCG protects against more than just tuberculosis was first described by Kalmats and Guran themselves. And they said here that we can judge its influence, that's BCG, on the diminution of tuberculosis and general mortality. And that's nicely illustrated by these figures, where they showed that the mortality against tuberculosis was decreased, as was all-cause mortality. So I'm going to end there. We'll have questions at the end, but I'm also happy for anyone to email me at that address. Thank you very much.
Thank you, Nigel. I mean, again, an excellent overview, comprehensive and succinct. The speakers are going to give us plenty of time for discussion, so I really appreciate that. There has been one question in the chat which could just get you to think about, from Sassy, and I guess I can paraphrase it, is when we start thinking about giving BCG a bit later, is it affected by previous exposure to TB or NTM? And maybe that's something we can come to a little later. Thank you so much. So the last speaker is Professor Jamie Tricas, and Jamie's with the University of Sydney. He's just getting a bio. He's a bacteriologist who uses a multidisciplinary approach to define immunity to chronic bacterial pathogens, and develops new treatments to control infection. So Jamie's a professor of medical microbiology and deputy director of the Sydney Institute of Infectious Diseases, and with over 25 years experience working with pathogens. He's been involved as a principal investigator on the Advancing Vaccine Adjuvant Research for TB contract, which is a 5-year program funded by the NIH to identify the most promising adjuvant TB immunogen candidates for clinical development and define immune correlates of protection. Jamie's also a colleague on the Australian Tuberculosis Centre for Research Excellence, which is funded by the Australian government. So thank you, Jamie, for coming down from Sydney, and we look forward to your talk on progress in TB vaccine development.
Great. Thanks, Steve, and thanks to Fiona and everyone for the opportunity. So what I will try and do is actually integrate a bit with what Nigel had spoken about, because there is some, I'm not going to say overlap, but I think there's some nice complementary points that I will make that will back up and maybe challenge a little bit some of the concepts that Nigel raised. So hopefully this works quite well. So I guess I'll start with the question that in our current times that someone who's interested in making TB vaccines tends to get asked quite a bit. And that question is, we have COVID vaccines and we got these pretty quickly. Why don't we have a new or a better vaccine for tuberculosis? And of course, Nigel made some great points around this, but I guess I'll address these more specifically in terms of the immunity to infection and the type of selection of the best type of candidates that we need to address this question. So I think the first point to make is, if you open any textbook on vaccines, you'll see something like this, that a vaccine, and we know that this is how essentially all childhood vaccines work, is you develop antibodies against the pathogen of interest. And in most cases, for many of the important childhood infections, these are viral infections. And we know essentially for most viruses that you need antibodies to deal with them. And of course, some other bacteria. In terms of tuberculosis, this is not necessarily the case. So despite the fact that there is some, I guess, recent evidence suggesting some role for antibody classes, what we do know, and Nigel touched upon this, is that we certainly know from most animal models and a lot of studies that it is T-cell immunity that correlates with protection to some extent, but specifically what type of T-cell immune response or what type of cellular immune response is required is not really known. And Nigel gave a nice example of, we all pinned our hopes on these T-cells making multifunctional cytokines that came up as very strong correlates in animal studies that haven't necessarily quite panned out in some of the human studies that we've now looked at. So I think it is true to say, although we always say this in our grant applications, that we don't have correlates of immunity and we need to find them, and you need to fund us to do this. I think it's true to say that our understanding of the exact parameters needed for optimal TB protection in humans is not well understood. I think the other important point that is maybe a little bit overlooked in some circles is that we also don't have a really strong understanding of which are the antigens that we need to protect against infection. So again, if you make the correlation with viral pathogens, and for many viral pathogens, for example, SARS-CoV-2, there's a very, very well-defined antigen that you need to target to induce protection. This is not the case with tuberculosis. So there's many candidates. We all work on our own candidates. There's candidates that are expressed at different stages of infection. And I guess one of the caveats maybe to what Nigel was talking about, in the sense that for some of the vaccines that have been tested in humans, or for some of the studies, we haven't seen strong correlation with immune responses, a particular type of immune response. That, of course, is a particular type of immune response to the particular antigen that was tested in that vaccine. So it may be different combinations of antigens, or even different antigens that we're yet to test may induce better protection, and it could be with a type of immune response that we haven't quite identified. So the whole thing about antigen selection is also very complex. The other point, of course, is that unlike, again, some of the other vaccines that we currently have, we may need to develop vaccines for optimal efficacy that are effective at different stages of infection. So ideally, we would like a vaccine that stops exposure, so a pre-exposure vaccine. But as we know, there are a large number of individuals who are latently infected. So it could be that a candidate vaccine that's able to stop progression from latent infection to active infection would be a useful product to have. The issue here is that we know that the organism Mycobacterium tuberculosis can have quite a different expression or makeup of antigens in the latent state as opposed to the active state. So you do have an issue there of antigen selection as well. And of course, the other area that many people are quite interested in is looking at some form of immunotherapy. So can we develop vaccines that we can use to treat people who are actively infected, or can we use these in combination with available chemotherapeutic agents? And so when we think about vaccine trials, and I'll talk about this on the next slide, we have different trials that looking at different things. So we can have vaccine trials where we talk about POI. So we're looking at the capacity of those vaccines to prevent infection, or vaccines that are needed to prevent disease. And there's different ways that we use these type of trials. So where are we in terms of tuberculosis vaccines? Well, this is I think a relatively recent overview of the tuberculosis vaccine pipeline. So you can see that it's reasonably well populated, although not as well populated as other diseases such as HIV, for example. And that's, of course, is probably an issue of funding as well. There's different types of vaccine products, and I'm not really going to go into this, but we're really looking. So again, I guess it's an important point. Do we have an optimal vaccine type? Well, not really, because we're testing live vaccines, recombinant vaccines, subunit vaccines. Now RNA vaccines are coming into the pipeline as well. So there's a whole different range of vaccine types. But if we look at just a few examples, and I think this will probably give us a really good overview of the vaccine landscape. So Nigel spoke about this. So this was the first vaccine trial, large vaccine trial after BCG, was the testing of this modified virus within expressing a single antigen. Nigel indicated it didn't work very well. So in fact, protection against disease was quite low, around 70%. Protection against infection was essentially zero. But I think the important thing to note here is that this actually led to a lot of pessimistic overview or understanding of where we were with TB vaccines for a long time. So the assumption was this was a relatively well-designed trial. It didn't work. Is that going to set us back a lot? And there's issues about that particular vaccine. And if you go back and look in the literature, you'll see there's a lot of commentary around, did the animal data really support the progression of this vaccine? And are we surprised it didn't work very well? Nonetheless, I think that was a really important study to show us that we can test new vaccines in the field and be able to do large efficacy trials. So there was a bit of a gap. So that was 2013. A few years ago, 2018, I guess, we saw this study. So this was a study which was looking at comparing two vaccine types. So one of them was a subunit vaccine, so a protein in adjuvant, and it was looking at protection against infection. So these were individuals who were previously BCG vaccinated, but were not positive in a quantiferon test. So they didn't have TB infection. And looking to see that if you gave them the new vaccine, or if you actually revaccinated them with BCG, what happened in terms of protecting against infection. The new vaccine was okay, but it wasn't great. The only candidate that actually we saw some effect was revaccination with BCG. So now there are new trials looking at the potential impact, larger trials of the impact of BCG revaccination. And that's maybe something that Nigel might want to touch on, because that's certainly his expertise. And I guess probably the most important, I would say, in terms of where we are in the field was this trial. So this is a trial of another subunit vaccine. So it's a vaccine in adjuvant that was developed by GSK, but progressed by GSK. And this was testing this subunit protein in people who were previously infected. So if you think back to my schematic where we showed people who are lately infected with TB, can we develop a vaccine to stop progression? And overall, what this study showed was that this vaccine candidate, the efficacy in stopping progression to disease in individuals who were previously infected with tuberculosis was around 50%. And what we saw was that it's probably not very useful in terms of a precise understanding of the immune response, but certainly what we saw is that the immunity correlated to some extent with persistence of CD4 T cells secreting these multiple cytokines, and surprisingly, actually not a high level of CD8 T cells, but this is probably also a function of the type of vaccine. So this, of course, is probably the best we've seen, and I think is a great outcome that we have a vaccine that works around 50% in terms of this population. The other thing I'll say very quickly, and I don't know if I have time to talk too much about this, but we're on this list, but we're pretty early. So we have one of our candidates, which is supported by the Tuberculosis Vaccine Initiative. And I'll just show a few slides on this towards the end. But I think the most important thing to maybe discuss a little bit further is this vaccine trial. And so it's interesting. So you may have seen this very recently, only a few weeks ago. Additional funding was released for this. So that vaccine trial that I showed you was a phase 2B in a few thousand individuals. What has now happened is that GSK had indicated that they would not fully fund a phase 3 trial for that vaccine. And if you look very closely, I've just cut and paste a few sentences from this Nature article, and this was essentially abandoned due to lack of commercial potential. And it was the Gates Foundation who essentially took up, or they're funding most of this trial, who are providing the funding for this candidate vaccine, and the Wellcome Trust. So they've put in $550 million to do a large phase 3 trial to determine the, hopefully show that we can replicate that high level of efficacy in a larger population. It's interesting though, because very recently the WHO released a report on the business case or an investment case for tuberculosis vaccine, and actually showed that all things being equal, there is $372 billion market over 15 years for tuberculosis vaccine. So I think seems like a reasonably good commercial potential, but it seemed like GSK at the time did not consider that the investment was necessarily worth the outcome. And again, Nigel mentioned some of the issues around tuberculosis vaccines and how they're perceived in terms of a potential product to be launched. But I think it's great that, you know, the community has got together to continue to fund this vaccine. So just for the last few minutes, I'll talk a little bit about correlates, and then I'll show you just some of the work that we're doing very quickly. So I think one of the most important studies of the last few years in trying to work out how can we develop correlates of protection against tuberculosis. So how can we avoid having to do large efficacy trials or doing a lot of early clinical trials to determine which are the best candidates to progress into the clinic? Can we have some form of correlate in animal models that will allow us to select this? And I think a really interesting article that came out a couple of years ago in Nature looked at the impact of intravenous BCG in macaques. Now, of course, you may say that intravenous BCG as a vaccine approach doesn't seem very practical, but it is very useful in allowing us to identify correlates of immunity. And what they showed in this model is when you give intravenous BCG to macaques, you essentially cleared the bacteria from all the animals. And this was much better than giving intradermal BCG or giving aerosol BCG. And what they're showing on these graphs here is just the number of bacteria that are present within the lung or the lymph nodes. And this is really unprecedented. For those of you in the audience, I'm sure there's quite a number who have worked on tuberculosis and BCG in animal models. It's very, very difficult to clear tuberculosis from an animal. Once it's in, it's almost impossible to eradicate with a vaccine approach. So of course the question is why did this happen? Probably the strongest correlate that they showed was that there was a very high number of T cells that were residing in the lung that were specific for the vaccine. So this is an important point when I show you the next few slides. So that's one particular idea. They also did show that it might be the presence of particular antibody types as well that correlated in a follow-up study. So this is giving us a few clues. So of course the question is, as we're trying to develop new vaccines, because we can't assume that M72 vaccine that is moving through is necessarily going to show fantastic protection in a larger trial. I think we need to keep populating the pipeline. How do we do this? So we're doing a few things in our lab. A few different approaches. I've got a few minutes left, so I'm just going to talk about one of them. So this is an approach where we're using a fusion protein. Again, we have our own antigens we're interested in, combined with a novel adjuvant. And this is the one that we have support from the European TB community. And what we're doing with this particular antigen is we've identified particular antigens, certainly ones that we've discovered in our lab, that seem to be very important for the bacteria to survive during the chronic state of infection. So these are antigens that are highly expressed in animal models during this persistent or latent state of infection. And we've combined these with antigens that were previously discovered that were seem to be very important in the early stages of infection, so secreted antigens that the bacteria makes early on. So we developed fusion proteins, and we've done a lot of work with these. What we've been doing more recently is looking at ways. We've also, have evidence in our models that really generating this strong immunity in the lung is very important. So we've looked at vehicles or different types of vaccines that we can use to safely deliver vaccines to the lung. And we've been using an adjuvant called Advax, which is essentially a polysaccharide, and it's shown here in green, and in red we've got our antigen, and these are cells within the lungs of mice. And what we've shown is when we directly deliver this vaccine to the lung, these cells just gobble up this vaccine. They love it. We don't see any negative impact on the animals. We see very high levels of vaccine in the lung. And I won't go into too much detail here, but we've done a lot of fairly detailed immunology to show that the vaccine gets into all different cell types. What's quite important, what we think is important, is that the vaccine is quite persistent and it likes to get into dendritic cells. So the purple here is showing cells that are expressing this marker for dendritic cells. And we know dendritic cells are very critical for initiating and generating immune responses at local sites. So we're seeing a high number of dendritic cells in the lung and in the lymph nodes taking up the vaccine. And what this has led to is us identifying that within these animals, and this is sort of the last data slide I'll show, is that we've been able to identify that these antigen-specific T cells, so these T cells that recognize our vaccine, are residing in the lung, so they're tissue resident. And interestingly, the major marker of protection that we find is the expression of interleukin-17, which is another important cytokine in the development of immune responses. And again, Nigel spoke about the fact that these traditional multifunctional T cells that make interferon gamma, IL-2, TNF, they do not correlate with protection in our models. It's these particular T cells making this particular cytokine that do. And this is just the animal study showing if you remove this cytokine, you lose your protection against infection. All right, I think I'm at the end of my time, so I'll just finish. Steve mentioned this, the other thing that some of this work has led into, and we're working with a number of different partners around Australia and also internationally, is to now developing a program where we're taking the information we've learned for different adjuvants and different antigens and trying to look for the optimal antigen adjuvant combination to progress as vaccines, as TB vaccines. Again, based on the fact that I told you, we don't have a great understanding of what's the best type of immune response and what are the best antigenic targets that we should be using. And just to finish up, a number of collaborators here at the university that we've been working with across here at the company Vaccine, and then a number of groups. And we're also obviously very grateful to have funding to continue this work. So thank you.
Thank you, Jamie. That's excellent. And time for some questions. I think Paul Wood asked whether there was a human challenge model for TB being developed at the Bill and Melinda Gates Foundation. I think that's Anne Ginsberg's group, but is that still ongoing, was the question?
Yeah, yes. From what I know, yes, but I don't know anything about the details.
Yeah, but Fiona's put a link in the chat that you'd be interested to look at.
Okay. Another question, if I may, to both Nigel and you, Jamie. You know, we use BCG in newborns who have immature immune systems. We know it doesn't work, but it has limited efficacy and duration of protection. And the group that we want to get to prevent TB are those who transmit, such as older adolescents and young adults. If we give it to young adolescents, do you think we'll get a better immune response and a longer duration of protection? What are your thoughts? Not necessarily just for BCG, but is that the target group we should be thinking of more?
I'll let Nigel start.
I think that's a Dorothy Dixon. Yes, I think, Steve, I'm sure you would be advocating that is the target group, because that's where the bulk of TB diseases and probably deaths is occurring rather than neonates. I would continue to argue that BCG is highly effective in that first few weeks of life against the kind of TB that babies get. So I think we shouldn't throw that out. But I think the real need, as you say, if you're going to throw your money at something, that's where you would be putting it, because BCG is of no use once you get to adolescence, or I should say limited use compared with its early. Now, whether that, the reasons for that are hugely debated, whether that is duration of protection or whether you need different bits of the immune system protect you as you get to those ages. There's lots of different theories. But yeah, that is absolutely where a lot of the efforts are going. As you know, BCG revaccination studies, which have not been as encouraging as had been hoped. But Jamie, I don't know whether you want to add anything.
Yeah, look, I think, look, the way that we think about this in terms of how we're developing, and I think the community is that optimally you maintain BCG and then you develop a boosting vaccine that you give at a particular stage, and it could be 10, 15 years when you start to see that waning as a boost. So all of our models that we set up are all based on boosting BCG. I think that probably the leading candidates to replace BCG, there's at least one attenuated vaccine that I didn't talk about that's moving into phase 3. And I guess the idea there is, will you develop greater protection than BCG using this attenuated strain, and will you maintain the beneficial effects of BCG? And you could argue that you probably would, because they're going to be similar pathogens, and they're going to maintain a lot of those similar potential mechanisms that BCG has in those non-specific effects. So, yeah, I think from our point of view, you maintain BCG and then you find a good boosting agent.
Thank you. Jim, you've asked a question in chat. You could speak to that if you would.
Thanks. I think you probably just answered it with the last part of your last answer. But I mean, given the difficulty it's taken to demonstrate and then accept the non-specific benefits of BCG, is any future vaccine being considered as an additional vaccine rather than a replacement? Or are they really thinking about replacing it? And then how would you go about demonstrating you're maintaining those non-specific benefits?
I guess you're right, Jim. There's been a lot of the vaccine effort is talking about boosting BCG, because I think quite rightly people are worried about stopping the birth dose of BCG and the potential consequences of that. Some people have advocated for another dose in the first year of life or in older childhood. I mean, the problem with BCG is, of course, there's a couple of other problems we haven't addressed. It's not the easiest vaccine to implement because you have to have people who are skilled at giving intradermal vaccination, which explains why a lot of babies don't actually get their vaccination at birth. They actually, unfortunately, don't get it till they turn up at a health facility for their first DTP. So there's some very simple things we can be doing to prevent deaths by just making sure that babies do get their BCG immediately after birth, as is recommended by WHO. And there's been a big push by WHO to take that. It's a very low-hanging fruit to use what we already have effectively. And of course, the second thing is the issue of scarring and keloid and the other kind of relatively rare complications of lymphadenitis, etc. But it's not, it's not the easiest of vaccines in terms of its effects. So giving additional doses is not that attractive versus some other better vaccine that boosts the initial effect.
Yeah, and I think that answers some of the other questions that are there as well, Nigel. Some follow-up questions about giving repeat BCG.
Thank you. Sassy, there's a question regarding from Imelda Manarong regarding the fact that I think you're suggesting that in your setting, community infection provides a large amount of disease and infection in the home, which is obviously in clear contrast to a country like Australia, where most of the exposure is identifiable through household contact screening. But in your situation, it's different. Do you want to speak to that? What you meant by more bacteriologically confirmed TB is coming from the community rather than the household?
Yeah, so this is a contact investigation performed by community cadres, so non-health professionals. So they were trained to do contact investigation and they visit patient house and then the neighboring area. So they have to investigate at least 20 individuals living nearby the index case. And in their study, they identified more sputum smear positive individuals or expert positive among individuals who live or who are not a household contacts. So yeah, so that's the result. And is there any characteristic or cultural? So this is a study in 30 provinces of Indonesia. So we haven't looked into detail for characteristic for each province, but it seems like the result is similar in most of the provinces. Yeah.
Thanks, Sassy. Paul, you've asked a couple of questions. Over to you. Or a comment.
I think it's a similar question.
No, this is more to do with registering vaccines for off-target protection. Did you want to speak to that, Paul? Or?
Yeah, I suppose just from a person who's done sort of vaccine registration work, I can't see how you could do a study that would actually include off-target effects, because the regulators, I think, wouldn't allow it. So as much as I can understand the desire to keep those benefits of BCG, I just can't understand how you'd ever get a vaccine registered with that additional effects.
Yeah, I agree. I mean, I agree. I mean, I think what will then happen is it'll obviously be ultimately on the on the overall efficacy against tuberculosis. And if that vaccine then becomes licensed, there'll be a whole lot of studies subsequently to see if there's any off-target effects, but keep Nigel in business. But I don't know, I honestly, BCG always surprises us. I think that you look in many different models and where you expect BCG to fail, it doesn't. And so I'm just not sure if an attenuated vaccine, which looks quite similar to what BCG does in the animal models, will necessarily be so different that it'll end up being a superior product. But of course, it needs to be tested, and it's good we've got these things moving through.
Great. Just coming to the last couple of questions, Nigel, you wanted to ask Sassy about asymptomatic carriage. Are you meaning subclinical disease, people who are transmitting but are subclinical? Is that? I'll leave it to you. Yeah.
The nightmare that it may be that those without any symptoms are the ones transmitting or playing a big part in the burden of transmission.
Yeah, and I think that's a massive challenge for case detection strategies in the community, and a reason why vaccine again is so critical, because it'll be able to prevent subclinical as well as clinical disease. But national TB prevalence surveys done in many countries frequently show that 50% of bacteriologically confirmed TB is asymptomatic, and that's a massive challenge. It's good to highlight that. Thanks, Nigel.
Can I just very briefly return to my commercial for continuing the use of BCG in relation to Paul's comment? You know, I totally agree. Even if licensure was allowed to use off-target effects, it would still be very hard, because with reducing mortality, the numbers you would need in those trials now are into the tens of thousands. So it would be very, very difficult to do. But also, just to remind everybody, just to kind of return to this, there's still so much we don't understand about BCG, and I didn't have time to go into it in great length, but there's quite a lot of other indirect evidence that different strains have different efficacies. And so I think we can do a lot more with what we already have, and that really needs testing. And we were about to do all that before this COVID thing hit. We were about to do a big study in Africa comparing different strains for both specific and non-specific effects. And to my mind, these need to go on in parallel, but we should not. And we should remember just how good BCG is and the additional potential there is for it.
And I know we probably have to wrap up, but just one really interesting study I saw, Nigel, I don't know if you've seen it, but there's a recombinant BCG that's coming through that was from the Kaufman group. And they did a study where they looked at immunogenicity. So in that vaccine, every single animal study compared to BCG showed superiority. In some of the testing they've done in humans, if you look closely at it, BCG is better. The immunogenicity of BCG, at least based on the parameters they're looking at, showed superiority of BCG in generating immune responses in humans. So I guess that's why I say you can never underestimate BCG. And I think to have a BCG-like replacement, I think is going to be hard. Or a better version of BCG, it's going to be a difficult ask, I think.
Thank you. I think we're going to need to wrap up. I did notice in the comments, Katie Dale, who's an epidemiologist with Victoria, said that there is actually evidence of quite a lot of extra outside the household transmission from whole genome sequencing work in Victoria. I guess my point was that as you move from a low TB endemic setting to a high TB endemic setting, there's more and more transmission we're assuming is going on outside the household as well. And certainly amongst adolescents, where there's a rapid rise in infection and disease in countries like Papua New Guinea or Indonesia, we know from social science that that's the most sociable time of our lives, and we have a lot of contacts in lots of different settings at that stage. So again, this talks to the epidemiology that informs vaccine. I might at this point, I think there's a comment about the dollars. It was pretty sobering what you suggested there, Jamie, about the commercial interests of a company. It wasn't that long ago, The Economist rated TB as one of the no-brainers for investment, a $40 return on every dollar invested in TB. One of the biggest no-brainers, that was by The Economist. But even these sort of information doesn't seem to sway the way we're moving forward. Fiona, I might leave it over to you now. I think it's been fantastic. So thank you from me, but I think I'd like to really interesting talks, a huge amount of information, but I might leave it to Fiona to wrap up on behalf of the group. Thank you.
Oh, no worries. Thank you very much, Steve. And really a big thanks to Sasi, Nigel and Jamie for fabulous talks. I've learned a lot. And so really thank you very much for your time and really interesting questions and discussion. And I really appreciate that. And great to see such a good turnout. And we're aiming, to do something similar every 3 months. So thank you, everybody. And have a good rest of the afternoon. Thank you.
Thanks all. Bye.
Thanks all again. Thanks.
Thank you. Bye.
Bye.